It's my great pleasure to welcome you for our second Knowledge Exchange seminar organized by the School of Biomedical Sciences at HKU Med. My name is Ralf Jauch and I represent the Knowledge Exchange team from our school. And we started the seminar series with the aim to communicate some cool and exciting science beyond our geeky research community in a way that it can also attract interest through social media and of course the actual lectures for other stakeholders, the community, people from other fields and from other, from outside our faculty and our immediate research area. And we do co-organize these seminars with stakeholders from other fields. So we had the first one co-organized with the German consulate, and today I'm very pleased that Tarani Chandola from the Sau Po Centre of Aging, representing the social sciences of HKU, will co-host the seminar with me, and he will also say a few words in a moment. So, of course, aging is a topic that affects us all, and we often view aging as a wear and tear, as an accumulation of damage that's kind of unavoidable and progresses, and there's nothing we can do about it. And of course, as we all know, aging is the root cause of most of the diseases people eventually die from, be it cancer, diabetes, neurodegeneration, cardiovascular diseases. You know, there are people in the field that put forward these bold ideas that we should stop worrying about all these diseases and studying all these diseases individually. We should much rather focus on eliminating the root cause of it, which is aging, and that would at the same time provide solutions for aging. This is of course a very controversial idea, but if you follow social media and if you look into what some of the thought leaders in that field are discussing about, that's certainly some of the bold minds actually trying to accomplish. And it's not a niche activity because some of the richest people on earth feel they have the resources to basically tackle aging. And we have a couple of the billionaires of this world, be it like controversial figures such as Peter Thiel, Jeff Bezos, they're pouring in money and resources in supporting this concept, and that we can actually defeat aging. So whatever you believe as a scientist, it is something that's backed up by powerful people with strong interests, and it's definitely a fascinating topic to discuss and to debate. And actually today's speaker, I will introduce him a little bit later after Tarani's short welcome speech, is actually very active in this community, and I invited him today to share his views and his hypotheses, his models with us. And when you actually search him on Twitter or YouTube, you will actually find a lot of social media activities. That's why I also thought he's great for a knowledge exchange seminar, because he's very active in this debate, and the maker and shakers in the aging community, they do know his name. And it's a controversial debate, sometimes a heated debate. My laboratory is also interested in this. You may have heard there is indeed a way to reverse aging on the level of cells. Yeah. Here at the medical faculty, many of us, we use this method in turning a skin cell, a blood cell, or urine cell back into an induced pluripotent stem cell. And this is used to study diseases, to make cells that we can actually use as a drug and give it to people to fix degenerative conditions. But during that process, we have magically also the rejuvenation of cells. So when we take a cell from a centenarian, we can roll back the aging and we make a cell that in many ways, all the features resembles an embryo. So this sparked a lot of excitement in the field, not just in the stem cell community, also beyond, that when we can reverse aging on the level of a cell, can we also do this on the level of organs or even on the level of whole organism? And there are serious, credible people in the community who think that is actually possible, and some of these powerful and rich people, they do invest in these endeavors, and with the hope that this can indeed be achieved. To fix organs, to repair damaged organs, but even to kind of stop or reverse the aging clocks. But that's it from my side. And before I introduce Peter a bit more properly, please, Tarani.
Thank you, Rolf. So it's my huge pleasure to welcome all of you and welcome Professor Lidsky to today's seminar. And before I start, perhaps a little bit of background on why I'm standing here. So my name is Tarani Chandola. I'm based in the Faculty of Social Sciences. I'm a sociologist, so not a typical person that might attend one of these seminars, but thanks to Professor Yauk's invitation, I'm glad that myself and other colleagues from the social sciences can be represented here. So a little bit of background about the Sau Po Centre for Aging that I'm a co-director of. So we think it's one of the leading social gerontology institute centers, not just in Hong Kong, but in the wider region. And actually, we just celebrated 25 years of our history together. So it's been running for 25 years. It's led by Professor Vivian Lau. So she's based in the social work department, and her research is very much focused on caregiving among older adults, as well as on robotic assistance for elderly people. So she's got these two strands of work that are, a key focus of the Center of Aging. But apart from her work, we've also got work on end-of-life care, both in Hong Kong and elsewhere. So it's a really important topic, given how old, the large number of older adults we have in Hong Kong. And we also have a lot of work trying to work with the government databases, trying to make sure that they link up to identify vulnerable older people in the community. So that gives you a flavor of some of the work that we do in this Center for Aging. So apart from Professor Lau, there are four other associate directors, and one of them, we're mainly based in the Faculty of Social Sciences, but one of them is based in the Faculty of Medicine. And we're so glad to co-sponsor Professor Lidsky's talk here today. I'm really very interested to learn more about his views on how we can stop aging and how we can treat aging. So thank you very much.
Yeah, but now without further ado, let's briefly introduce today's speaker. And I'm quite impressed. He even brought his own social media team with him, so which is a new experience and very good for us, because we also are planning to promote this beyond this seminar, this type of seminar. Peter Lidsky is originally from Russia. So he graduated from Moscow University in virology before moving to Switzerland, to the ETH. And then he moved to UCSF, and he's been actively involved in aging, but also virology research. So he used animal models such as Drosophila, fruit fly, for his research. And he just recently got recruited to the City University of Hong Kong, and we were introduced by a common friend in the US who basically, he was looking for someone who helps him to settle in Hong Kong. So some connection, also teach him about how to deal with fruit flies, which I don't do anymore, but whether I could connect him. And then when I met Peter and we also brainstormed some ideas, then I also started to follow him, and then I realized how prolific he is with his outreach, that his ideas are very well known. He is involved in the global debate on what aging is really about, how aging works. And he has a very staunch following here. Also, he gets involved in many of these debates and controversy. And so when I learned all this and his thought-provoking idea, I thought he is a perfect speaker for this type of seminar series. And I'm really looking forward that he will inspire us today with his ideas, what aging is all about. Peter, looking forward to your talk.
Thank you so much for having me and for this kind introduction. So, today we will discuss aging and these non-orthodox ideas. I was asked to introduce as much social component as possible, So, and I will start my talk with the little opinion poll. First of all, raise your hand if you want to live forever. Okay, not many. So if you want to live forever, if in good health. A little bit more. So, and the third question, do you want to stay healthy until death? Almost everybody. For those who didn't raise the hand, we can rephrase this question. So do you want to die with cancer? Do you want to die with Alzheimer? I don't think anyone will raise the hand. So it's a problem that concerns everybody. If it will go as it goes, then all the 8 billion will die, and many of them will not die gracefully. Little problem that we have is aging. So as you know very well, that probability of dying with almost every disease is increasing with age. Seems to be that the problem is huge and should be in the center of social interests. And let's look how we fund this problem, because if it is so big, then it should be funded properly, right? So this is in NIH budget before the American Baraboom. So you can see that the Aging Institute scores 9% of the total budget, which is not that bad. So it's still less than infectious diseases, 14, or cancer, 15, but 9% is still a number. However, if you look at the breakup, fundamental aging research accounts only for 8% out of this 9%. The majority of funds are going towards Alzheimer, which is just one of these uprising curves. So why it is so? Recently, there was a paper by J. Olshansky who showed that actually we exhausted our potential to increase life expectancy. Life expectancy at birth has been raising for last 100 of years, and now it's plateauing or even going down. So, and to increase it further, we need to make some medical breakthrough. This paper has been commented by Eric Worden, one of the key people in the aging field, the director of Bach Institute, who wrote that this study probably give pause and ponder the enthusiasm of those claiming that radical life extension is near. We should instead focus on the tasks at hand, improving health span. Meaning that instead of targeting the common reason for all these curves raising up, we need to target these curves individually and in the end improve health of aging people. So I have met him like a month ago and asked him what did he mean with this, and he said no, you didn't understand me properly, so I'm totally up for fundamental aging research. We need to find the mechanism, the cause, but we need to report some progress to our funders and the communities. And for this, we need some incremental results which are not possible to achieve like in about the fundamental cause. So we need to target some age-related diseases. So it seems to be that the basic aging research is very difficult. I want to introduce you to another scientist. It's Dr. Miriam Grunewald. She is actually not an aging researcher from the beginning. She was working on vascularization, like how the vessels grow. And she have had mice, this VEGF, this is vascular endothelial growth factor mice, that have this factor twice higher than norm. And actually, she forgot some cages in the vivarium, and then she came back disappointed because it costs her money. So she realized that the experimental group is visibly younger than control group. And then this is a paper in Science, and the interventions was very effective. So not only the average lifespan increased, that would be like this, but also maximum lifespan increased. And for a long time, this intervention was one of the best genetic interventions in the field. So, and there is some controversy here, right? From one side, mainstream top scientists believe that good interventions are very difficult to do. On the other hand, scientists from neighboring fields are developing interventions just by being accurate, sometimes forgetting cages, but being curious about what came out of this. So, and let's ask ourselves, what is the main bottleneck in aging research? So, my belief is that the main bottleneck is that we don't know what aging is. So if this is a paper led by Vadim Gladyshev, he actually did a survey of different experts in the field, what is aging, and they all give different answers. So the best he could do is to just build these clouds of words, and you see the questions. How do you define aging? Well, no, something is declining, something bad happens with people as they age. What causes aging? Something damaged, yeah. How do you define the rejuvenation? No, then it goes somehow back. What is the most important unanswered question? This is aging, actually. So, and if we approach the questions stringently, gerontology as a field doesn't exist because the main object doesn't have scientific definition. So, and we just don't have a common language to discuss the issues. There are some people, some labs that are trying to develop this language, the paradigm. We are one of them. We're also working on our Esperanto, and some of the people are coming from mechanistic side, some are coming from evolutionary side, and we more tend to evolutionary ideas. Why evolution? This is Theodorus Dobzhansky. You all know this quote very, very well. So that nothing in biology makes sense except in the light of evolution. Because our bodies were shaped for billions of years to spread our genes. And this gives an incredibly powerful tool for understanding. Every phenomenon that we want to find a mechanism, we typically use evolutionary or ecological model to find this mechanism. Now, let's go with some examples. For example, the paradigm of infection is colonization of an organism with parasite. Having this in mind, we go study parasite replication, we study immune response. With this, we find antibiotics and vaccines. So, or diabetes that we eat sparsely, but we need to stabilize the sugar level, and this leads us to discovery of insulin. So, however, for aging, this model hasn't been built, I believe, and that's why we are missing the mechanism as well. So, let's now look at the ideas about the evolution of aging, the historical ones that have been proposed so far. The first one came from Haldane and Medavar. They suggested that animals just do not survive to the age of senescence in the wild. So if nobody survives to the age or to the old age, then to this age, then for evolution it doesn't matter what happens with an animal, and aging therefore is just a neutral feature. However, this is factually not true. If you look into the animal populations, for example, this population of lions, you see that probability of dying in the lion of age 14 is much higher than in lion of age 5. Meaning that some other factors should be involved to explain aging. So in this situation, a long-lived lion that has not that high mortality would have evolutionary benefit and will outcompete the faster aging lions. So to deal with this problem, George C. Williams suggested his antagonistic pleiotropy model. He hypothesized that there are some genes that are beneficial in young years and detrimental in old years. And so this beneficial function helps a huge young cohort. Detrimental function damages a tiny old cohort. And in the end, net benefit of this gene is positive. However, there are no good examples of such genes found since like 1957. So there are genes with the opposite functions, but this is not enough. So also there should be situation and evolution cannot disconnect the beneficial and detrimental function. And these examples are basically missing. So the next idea came from Thomas Kirkwood, who suggested the existence of fundamental trade-off between lifespan and reproduction. His argument was that we have a limited amount of resources like nutrients, energy, and we can invest this energy into either repair or reproduction. And with this rapidly aging but rapidly reproducing variants can outgrow the long-lived but slowly reproducing variants. This model has some mathematical problems that I will not speak about today, but it also have clear biological issues. So, for example, caloric restriction prolongs lifespan. If we starve animals, they live longer. If we overfeed animals, they don't live longer. The other way around, get obese and die faster. So males are aging faster than females in most mammalian species, while they invest clearly less energy in reproduction. Why it's so? Food consumption decreases with age. The model predicts it other way around. If an older animal found food, and it should eat it and rejuvenate, and it never happens. So also poor ecosystems like Arctic's caves or deep ocean, they are full of centenarian species. And it's clear that to save energy, longevity is much better strategy than over-reproduction. So that's why the evolutionary mindset is not very not very widespread in the field. Many people say they are not interested in evolution. They have their own movie going on, and they are trying to approach it from mechanistic point of view. If we find mechanisms, who cares about evolution? The problem of this approach is that the biochemical systems are extremely complex. So this is a simplified scheme of what happens in the cell. And very easily one can say, okay, aging happens because of something in this corner. Then you will come to me with your experiment and say, see, the ends do not meet the ends. Then I easily say, that something in this corner happens because by whatever, and through 35 intermediate stages affects aging. So the freedom, the degree of freedom is so huge that a non-controversial theory of aging is possible to build around any gene, any process in our organism. And so there are hundreds of these mechanistic theories. And they are not contradictory, they don't contradict to each other, but they are very difficult to reject. So you know the Harman's oxidative hypothesis of aging took like couple decades to be found wrong. That's why evolutionary paradigm is essential. So that's why we are tolerant. But if we introduce another PC, so we fly quite far away from others. So because everybody thinks that aging is somehow an entropic process, damage accumulation, and we believe aging is a program. What is this dichotomy and why program? So there are two ways of thinking. So one way of thinking is that aging is about accumulation of damage in the animals of human body. Another approach is that aging is a programmed process. Some genes in us are encoded to kill us by the given time. So these are two completely different approaches, because in the first case, we need to try to make these dots go back to their places, or at least stop them from flying away. In the second case, we need to find this gun and remove the bullets. So in one case, we need to repair something. In another case, we need to break something down. These are two completely different projects. If we invest in the wrong one, we lose all the money, time, and motivation. So why most people believe in entropy? So it is supported by these old flimsy models, which I think people accept with big reservations. Entropy is not proved experimentally. Every experiment that is suggesting damage can be rationalized using the programmatic model, but historically, there were two very strong arguments against the program. So these two arguments were considered to be killing ones. And so the first argument is, what is this program good for? Because idea of evolution is reproduction. If I die of aging, I cannot reproduce anymore. And second, if aging is in our genes, then we need to see immortal mutants. If you treat our flies or worms with mutagens, we easily find flies without wings, without eyes. Why we don't find non-aging flies? So and these were considered killing arguments, and everybody believe now in entropy. So I have built a model that can resolve these two questions, and now I will present it to you. Let's start with the first one. Can death be good? So it seems to be impossible because we need to reproduce, right? However, in this very convincing black screen, there is one little white dot. This is a case when death is accepted to be something good. So this is so-called ABI system in bacteria. It stands for abortive infection. And this is a suicidal mechanism that if bacteria is infected with a phage, then it is committed to die. It cannot clear it. So, but before dying, it will produce a lot of viral particles. And these viral particles will infect its brothers, which is also good, also not good, because this, they share the same genes, basically. So, and protect its skin, this doomed bacteria kills itself. The mechanisms are very well described. There are papers, so the phages are trying to inhibit this mechanism, bacteria are trying to invent new or overcome phage effects. So it's a conventional part of bacterial immunity. Can something like this happen in animals? Yes, it can, potentially. So let's say an animal is infected with a chronic sterilizing pathogen. What does mean sterilizing? Means that the animal cannot produce progeny anymore. So in human populations, such diseases are typically sexually transmitted, like HIV, gonorrhea, syphilis, and toxoplasma. They don't kill very rapidly, but they reduce fertility or sterilize completely. So second, it should be chronic, that there should be no recovery. There is no chance it will be healthy again. Then, actually, this animal producing infectious particles, it can reproduce still, but often this progeny will be also infected and useless. And so then it will probably die without leaving much progeny. But on top of being useless for evolution, it will probably it probably can infect its relatives, which are healthy, right? So then its inclusive fitness, how it's called, is like a value for evolution can become negative. And then evolution may want to kill such infected animals. But how to identify them? Idea is the age is a predictor for chronic infection. This is a picture from an experimental one. So you know the fish, like Arctic char, and its parasite. People collected fish of different ages, and they found that the probability of being infected increases with the age. And you see by the age of 8 years, 100% of fish are infected. Right, so there is no recovery in the system. Probability of being infected is constant year by year. very easy. So if this parasite, I think this parasite doesn't sterilize, but let's say it sterilizes. Then by age of 8 years, these fish are probably damaging for dissemination of their own genes. And evolution wants to get rid of them. So we could formalize all that with the mathematical modeling. For this, I developed this viscous population model that animals are express sort of territorial behavior. They are moving on the surface of this experimental field, rejecting each other. Why was it important? Because you remember that it is not only infection is key, but also that I need to infect my kin more than my non-kin. So this is the tragedy of the commons. If I kill myself to save all of you in this room, you will tell me, thank you, Peter, but my altruistic genes that forced me to commit this suicide will be lost. So I need to kill myself only to save my own progeny. And to model this properly, we need these viscous population models. Actually, the territoriality is a very common effect in nature. It's exemplified by these wolves in Minnesota, and the model recapitulates two very important and overlooked properties of animal populations, that animals often disperse from densely populated areas to sparsely populated areas. It's called the sink source model. And second, that relatives are interacting more often than non-relatives and infecting the relatives more often. So how does the epidemics look like in this model? So here yellow are short-lived animals, red ones are long-lived animals. Let's run epidemics there. And you see that the long-lived individuals are much more infected, because just they spend much more time in infected stage. And short-lived are dying earlier and remove infection with that. At the same time, in the absence of pathogen, if we mix long-lived and short-lived individuals, then long life always win, because they live longer, they leave more progeny, so they can easily displace short-lived. However, if we run this competition, it's a little bit wobbly, it shouldn't be like this. But yeah, if we run this competition in the presence of pathogens, the long-lived individuals in the beginning are starting to be expanded, but then they get increasingly infected and get displaced by shorter variants. And so this is the answer to the first question, that shorter lifespan helps to control epidemics of chronic sterilizing diseases. Okay, so we figured out the first question. Now what about the second? Why don't we see immortal mutants? In principle, we see patients that we can consider gain-of-function mutants, this progeria, right? But we never see this non-aging Voldemort. So why is it so? So, and for many classic gerontologists is like a final argument, the last nail in the coffin of the adaptive aging models. But, hold my beer. So I found an argument. There are actually, this pathogen control theory provides two arguments to resolve this question. One of them is here. So first of all, so the immortal Voldemort is possible. It looks almost as charismatic as original Voldemort. Have you heard about the naked mole rat? Right? So this is an underterran rodent that almost doesn't age. So its mortality doesn't grow with time, like for decades. The record, I think, is 30 or 40 years, while it is an animal size of mouse. And in the field, it's considered to be negligibly aging. So these rates can be neglected. And of course, these rodents have common ancestor with squirrels and rats. And along this branch, at some point, an aging mother should have give birth to a non-aging child. So this mutant is in principle possible, but by some reason, these mutants are very rare. And so the question, why? So the answer comes from another exotic system. So you know, in eusocial insects, there are queens that lay eggs and workers that serve there. And the queens are living way longer than workers. The difference can be 50 or 60-fold. So the termite queen is one of the longest living insects and who can live for like 5 decades. And actually, same situation is in this species of ants. And in this paper, authors found that some parasites can make workers that live typically short, make them as long-lived as queens. And for parasites, it's very obvious what is it needed for. This parasite infects an ant. It wants this ant to be eaten by a woodpecker, and where the adult stage of the parasite will develop. So it doesn't want the ant to die, because then everything is lost. It wants it lives as long as possible. And paradoxically, this is true for any other pathogens. Any other pathogens with chronic, that cause chronic disease, chronic infection. So paradoxically, pathogens are our allies that want to extend our life. So, and the idea, the explanation is that pathogens are constantly trying to prolong the host lifespan. Within this logic of host pathogens arms race, hosts invented security harnesses that prevent pathogens from manipulating aging, for inhibiting aging. And these security harnesses may protect aging mechanism from not only from manipulation by pathogens, but also from mutations and, unfortunately, therapies. So, and just for your curiosity, the same beneficial effects of pathogens are also observed in humans. So there was shown that some of the nematodes, they can sort of normalize the immune system, improving health status and even decreasing mortality. So the research is ongoing how to use this knowledge to produce better immunomodulators. So of course the benefit here is not comparable to what we see in ants, because we don't have this alternative program that allows us to live 50 times longer. So our plasticity is much smaller than in ants. So with this, I think I have answered 2 key questions that were initially raised against programmed aging. So we know that I believe aging evolved to control epidemics, and we don't see mortal mutants because of this security harnesses thing. So I think that there is no good reason anymore to believe in entropy as a cause of aging. So generally, it's a huge claim, because now if we go to some general conferences, for example, aging goes in this group of physiology, metabolism, nutrition, and that stuff. So if my model is correct, then it should go with immunity, infection, microbiome. So, and of course, and what can we do with this model? So what we are trying to do, we are trying to make work that would change mindset of people. And we are trying to develop interventions based on it. So first I will touch the also sort of social part, that changing mindset in such a field is very difficult. So this is Pedro Magalhães, who at some point drew a sketch of all the types of papers that are around in gerontology, like Virentir is bad, they think the measured is correlated with aging, so like stereotypical titles, and I take this on my account, actually, and that all aging theories, all previous aging theories are fatally flawed. So, and of course, people hate it because then you come to them, Luke, I'm your father, you know, it never worked. This is a huge difficulty, and that's why I invest quite some time and effort to communicate my ideas in alternative ways like social media, etc. So, what we are trying to do to convince people, there are many strange things happening in nature that are badly explained by classic models. Rats are living 5 times less than squirrel. Well, it is a very similar animal. Rat maximum live 5 years, squirrel 20. Everything what flies lives very long, like a sparrow can live 20 years, a tiny bat, size of the mouse, can live 40. Why it is so? So I told you that the queens are living much longer than workers by sharing the same genome. What is the reason why workers cannot live for also for that long? It's not clear. We touched already the non-aging mole rat. On the other side of the spectrum to the nearly immortal animal are these simiparous carnivorous marsupials and Pacific salmons that actually kill themselves after reproducing. And this killing is not aging. They die because of implementation of some hormone-based program. But what is the ecological role for this? It is not known, and actually, unlike classic models, this is a table from our review, this pathogen control can pretty well explain or rationalize all these phenotypes. So like 2 words about the explanations. So why flight can be related to longer life. So you remember that the kin-specific infection is very important for the model. So, and if infection is less kin-biased, then longer life is expected to evolve. And actually birds have 10 times larger dispersal, meaning that after hatching it flies away and found its own nest 10 times farther than a mammal of a comparable size. So why queens in eusocial insects are living so long? Because they can they comprise very tiny proportion of population. This is epidemiologically relevant. Now, how do we explain near immortality of the naked mole rat? Here it's a little bit more complex. So we believe that mole rats reinvented adaptive suicide, similar to what bacteria has. So that actually mole rats are also eusocial. They have queen and workers, and we believe that this is associated with this ability, evolutionary opportunity to use this adaptive killing of the first infected individual. If the very first infected animal kills itself in response to any infection, then the colony of mole rats can be kept sort of free of pathogens. And if we believe that aging is a pathogen control mechanism, then it may become redundant. So the reason why non-eusocial animals cannot do that is because in mole rats, due to eusociality, reproduction is disconnected from infection. The workers kill itself. Funny enough, there are papers that demonstrate that mole rats are extremely susceptible to some virus infections. And in this paper, people actually tried to put the gene of interest into the mole rat with using herpes simplex virus, and all mole rats died. In the other one, there was a spontaneous epizootics in mole rat colony that killed all of them in 2 days. Like a month ago, I also met Vera Garbunova, who is running a mole rat colony in the University of Rochester. And she said that they recently, not very recently, a couple years ago, they have had the same thing. So like all the mole rats died in 2 days. So, and idea of this work is to produce models to rationalize these observations and then produce predictions that we can test experimentally, like collaborating with people working on non-conventional animal models. So you are welcome to revise, to look at our recent preprint. This concerns rejuvenation. It's also a big paradox, because some animals in nature can rejuvenate, but so the examples are famous jellyfish, comb jellyfishes also do this, and some eusocial insects can rejuvenate, it seems to be. But by some reasons, they do it only under stress. Under optimal conditions, they age and die like we do. And question why? So the conventional models cannot explain that, because conventional models are still assume that death is something bad. And if there is opportunity to avoid death, this opportunity should be used. And so we could like prove it as a theorem and demonstrated rejuvenation. Even we believe our logic is correct. Even like rejuvenation of humans is not compatible with classic models of aging. And our pathogen control theory can rationalize the things and stress that rejuvenation is methodologically possible. So with this, I finished with the theories, and let's now discuss, let's now assume that pathogen control theory is correct. How would it affect developing of the drugs and interventions? First of all, if aging evolved to control infectious diseases, it's part of the immune system. The immune component in aging is increasingly recognized. Last summer, I think, this summer, the whole issue of the Nature Aging was featuring immune aspects of aging. So people already going this direction. However, nowadays, they consider the changes that are happening in the immune system to be sort of declining function and deregulation, something really not specific. So if our model is correct, then it predicts the emergence of some function that purposefully kill the individual. So actually, older we get, more immune system try not to protect ourselves, but protect our relatives from the infectious diseases that we have collected during our life. So, and this gain of function thing is not very well recognized by the community, that there should be something emerging by purpose, programmed, that we need to remove. At the same time, such examples are there. So here I want to advertise you other people's work. This is from Maria Mittelbrunn lab. This is from Laura Niedenhofer lab. So that actually if people specifically age the immune system, then they see accelerated aging not only in the specific immune cells, but also in solid organs. And they see strong acceleration of systemic aging in this system. So these are like, this is a control mouse. This is a mouse with aged T cells. So, and if the pathogen control model is correct, then it is it. So then instead of putting money in Alzheimer, we need to put money in rejuvenation of the immune system, because this can be probably the most effective intervention that can improve people's health. So, and again, it's a gain of function effect. It's not a loss of function. So we are collaborating with Matt, and I also discussed things with Maria. So, but I am not an immunologist, so we need to build this expertise to like make substantial contribution in a reasonable time. In the long term, we want to move this direction. However, we, with our expertise and resources, we want to resolve another interesting aspect of aging biology. It is cellular senescence. What is cellular senescence? Question number one. We also don't know very well. But what we know, that cells, if their telomeres are getting shorter, you know, telomeres are caps on the ends of chromosomes that are getting shorter and shorter with every division. If their telomeres are getting shorter, or if they're exposed to DNA damage, to virus infection, they go into this strange state called senescence. Typically, senescent cells are cleared by the immune system, but if it doesn't happen, they don't die, they're metabolically active, they sit there and produce some secretory molecules, and these secretory molecules create an inflammatory environment, can make other cells also senescent. It's called secondary senescence, and accumulation of these senescent cells is very bad for any like health condition, leads to metabolic disorders, cancers. Also, I think they're involved in neurodegenerative diseases. But what for is it needed biologically? Why did these cells evolve? Isn't clear. So if we go to the site of this ICSA, this is a International Cell Senescence Association, a very good community. I'm also a member of it. And this is actually the old web page, I believe. Let's study the definition of cellular senescence together. Cellular senescence is a programmed state of stable cell cycle arrest that is accompanied by a complex phenotype. Okay, this is observation. Senescent cells play a role in physiological processes such as tumor suppression, wound healing, embryonic development, whilst paradoxically they can contribute to aging, cancer, and age-related diseases. What does it mean? So it's clear that nothing is clear, right? So the cancer, for example, goes twice here, so because senescent cells were believed to contribute to cancer prevention, but later on appeared that their accumulation induces cancer. Also, wound healing. Wound healing doesn't increase with age. So, and what for these cells are needed isn't very clear. So people believe that if they sometimes cells should be removed, and this is good, but why don't they go into apoptosis directly? Why, if they are not cleared by immune system, they need to sit and do strange things? It is not clear, and some people call senescent cells zombie cells. That it should be dead, but it is not. It is roaming around, doing strange stuff. So, however, there was a small paper published 10 years ago, almost 10 years ago, that cellular senescence is an antiviral defense mechanism. And this paper was largely ignored, because there were some small shortfalls that people don't really take, and the claim is big. So, but we, armed with our pathogen control theory, we were very excited about finding this, and now we are testing that and expanding this idea. We call it immune militia hypothesis. I will tell you in a moment why. So, idea is that a healthy cell can be infected by the virus, then it produces cytokines, the signaling molecules that protect healthy cells from being infected with this virus. However, this cell, it can die, or virus can switch off this secretion, and then this supply will be exhausted, and cells, healthy cells can be reinfected. However, if these cytokines can make other uninfected healthy cells senescent cells can develop like resistance to viruses, secrete cytokines, and with this they can buffer the supply and create local inflammatory environment and like local memory about infection. So, and we call them immune militia because they are like militiamen who are recruited to the duty from any other job. So, and what very first experiment we did, we took A509 cells, very basic, induced senescence, and infected with a GFP-labeled virus. And you see it's very clear difference. Senescent cells barely can be infected. So we tried some other viruses, and here you see like 5 viruses from 4 different for different families. So they all are, they all hate senescent cells. We also have had SARS, it's not in this picture. So now, is SASP, this secretion of senescent cells, protecting healthy cells? Yes, it does. So if we take supernatants from infected cultures, we put them on healthy cultures, then we infect with viruses. Viruses are not happy, meaning that this link is probably also working. The cytokines provided by senescent cells can protect healthy cells from being infected. Next, to confirm that, we have inhibited one of the pathways, so-called interferon-1 signaling pathway. I skip a lot of intermediate stages, just bringing you to the final conclusion. So if we disrupt some key elements of this pathway, the receptor of interferon type 1, if NAR, or if we knock out the key component is immediately downstream from this receptor, we can rescue virus infection in senescent cells. So these are senescent cells, they don't have any GFP-labeled viruses. And if we take out these two proteins, you see that we start seeing some cells infected. Same happens if we put a chemical inhibitor of this pathway. So it seems to be that it's true that senescent cells are resistant to viruses not because they are zombie, not because they are weak and sick and viruses just cannot replicate there. It's because they are specifically immune. So, and the second component of the model is that the secretion of the infected cells should make other cells senescent. So this is still ongoing, but we have a principal result that indeed on day 2, now a virus is labeled in yellow, and this magenta is P21. So you see that cells that are sitting next to the infected cells, they indeed start expressing the cellular senescence marker. So with this, we have confirmed the 2 key components of our scheme. So that we believe we are on the right way. So the next stages will be like to find, we have clinical models for this, like in the virological context, because my aging theories are too non-orthodox, and I want my grants to go to the virologists. So that's why this will be sold as virology. So the next stages would be to study secondary senescence or study communication between senescent cells and immune cells, which is also like in the context of the immune militia, militiamen should be like coordinating with professional soldiers. So, and with this, I come to the conclusion. So the take-home message is that if the pathogen control hypothesis is correct, then aging is a programmed adaptation, and we need to inhibit this program and not repair damage. Second, aging mechanisms must have deep connections with immunity. Immune system is predicted to contain mechanisms that evolved to damage the organism, to limit the replication or transmission of pathogens. And third, the cellular senescence might be an antiviral mechanism. So the zombie have very good purpose in their real life. So with this, I want to thank all the people who worked for me. This is my lab at CityU. These are people who worked for me when I was at UCSF. So the work has been funded by some of the Raoul's grants. That's why he's here. So I have had 2 genius students working for me. Jing went to Yale, and Jacob went to Harvard for PhD programs. So also I wrote a book. So many of you who know Russian can buy it. It's in pre-sales already. So the English one will be out probably in October. So, and you can follow me on Twitter, and I'm happy to take questions.
Okay, yeah, wonderful thought-provoking talk, Peter. And what I really like is also this whole, maybe we bench scientists, we often forget that normally it's good to come up with a hypothesis, try to really examine what are the main thoughts, the main competing models before diving in. Experimental inquiries, that just reminded me on that. So maybe I kick off the discussion. But all the damage is happening, right? The accumulated damage in our cells. I mean, our heart cells, our brain cells, they're basically there for the whole of our life. And of course, they are suffering all kinds of damages all the time. So if we discover ever that program that might stop aging, it still would have to repair all these damages, no? Or do we need to inhibit this program right after our birth? I still don't get it. I mean, you're not doubting and questioning whether these damages occur. The DNA damages, the telomere shortening, the proteostasis issues, all that stuff. It's happening. So, but we don't need to repair it if we discover the program of aging?
No, so there are two questions in one. First of all, damage is of course there, but the the problem is that damage also there in any other diseases. So like in COVID, if you take a patient, lots of damage. Diabetic patient, tons of damage. So do we need to repair all these damages in COVID people? No, we typically vaccinate them just before they get sick. Same with diabetes. So we prevent this to happen. If indeed they already like far away in the disease, then yes, we need to repair damages. I don't think that if we switch off the this adaptive aging program, then the patients who lost their teeth will have new teeth outgrown. No, of course, this we would not be able to compensate. So I don't think that presence of these damages is an argument to believe it's a reason. So probably these damages are secondary to something else.
Mm-hmm. But then if we would know the handle, the switch to turn off the program, would we need to do this at the beginning of a person's life, or could we do this anytime, or would it be like a genetic modification? Can you speculate?
Well, I think we can probably revert aging from late stages because, reprogramming works, right? So we can reprogram cells. So likely if we rejuvenate the immune system, it will have a systemic beneficial effect.
So, next, Moride model is interesting. So they can obviously live quite long. So they don't see lights, as you mentioned, they are extremely sensitive to certain viral infections. But I would imagine that they live in the wild. They must be, exposed to all kinds of other viruses. So the viruses we don't know. Yeah. So the example you show there is something probably they have never encountered before because obviously that's not the virus. So. Another potential model relevant to your argument is that is bats, right? Bats actually they can also live quite long, but they carry a lot of viruses. So they have some certain levels of inflammation going on at a cellular level. So these two models, do you think they are kind of consistent or not consistent? Based on your theory,
they're consistent.
Your hypothesis,
yeah, the consistent idea is that bats are interacting extensively. They live like big groups, but the interactions are not as keen biased as in mole rats. So in mole rats, if the infected worker is getting infected, then it kills itself to save the queen.
So in the bats, because they are also carry a lot of viruses, they're supposed to be infected, but they're supposed to have a lot of, senescent cells. So, but they live quite long.
Yeah, the logic is other way around. So since bats have this very high dispersal, they cannot, they don't, the aging in them, a shorter lifespan in them is not that beneficial. because they cannot help their relatives too much with shortening their lifespan. That's why they evolved long lifespan. Since they evolved long lifespan, they have a lot of viruses, because viruses benefit on this. Since they have a lot of viruses, they also evolved bizarre immune systems to deal with this problem.
Okay, my second question is you see, we don't have the immortal mutants, but we do actually at the cellular level. There are tons of human cell lines out there. So many of them have normal genetics, components. They are not genetic mutant cells. They can be there for quite a long time at a cellular level. So that means we can have immortality at a cellular level, but not as an organism. So that's a difference between the cellular senescence and aging, I guess. Probably you can comment on this. Thank you.
No, yeah, the immortality of cell lines is rather supporting my model, that in principle it's quite easy to get cells immortal, and there is no fundamental rule that kills living matter after a certain time. So there is no contradiction. It's only support from this observation.
So yeah, thanks for the next talk. Are these aging models, are they necessarily mutually exclusive? Like, how does the pathogen control models explain the phenotype that we see in progeria and other diseases where there's a DNA damage, repair genes mutated, and the patients exhibit the aging phenotype?
Yeah, so the DNA damage probably play a key role in antiviral defense. So because many viruses that integrate in the genome are inducing DNA damage, and also viruses that do not integrate into the genome, they also induce DNA damage, probably via some innate immune response mechanisms. So generally DNA damage is probably accompanying the innate immune activation. And if the organisms, this is a part of the work that was not presented. If an organism feels that it is infected, it can be evolutionarily beneficial to accelerate aging. So because, we realize we are infected, maybe it's better to kill ourselves. So the model is more complex than that. So we need to be sure that this is not flu and not like common cold that would be gone in a couple of weeks. So, but if we are exposed to inflammation for a long time, we won't accelerate aging. And this is the explanation why chronic immune stimulation can lead to accelerated aging phenotypes. So the model is that in progeria patients, this constant DNA damage induces innate immunity and leads to acceleration of aging.
That's very nice theory. Different animals, or if you count plants, different organisms have different lifespan. So if it's programmed, why they are programmed at different lifespan? And according to your theory, what's the explanation?
Of course, there are many factors that affect optimal lifespan. So, for example, one cannot be a whale and live for 3 days because just time is required to grow and to use this size to propagate. Generally, from all the models, it's predicted that longer lifespan is beneficial. However, our model produces the selective factor that favors shorter lifespan. And we believe that the actual lifespan of a species is decided in a balance between these factors that favor longer lifespan and the pressure put by pathogens. So in this pressure from pathogens can include abundance of the pathogens in the environment, include population structure extensively, because as I will reiterate, it's very important that relative animals are interacting more often than non-relatives. We believe that this balance is individual for each species, and we are trying to describe some unusual situations like bats or mole rats that are clearly outliers. Did I answer your question?
Well, maybe not, but
No, if the question was what factors define the lifespan of the species, right?
yeah. So
So we believe that this is this lifespan are defined in the epidemiological models, so that how the chronic pathogen infections are running, how many pathogens are available, how the population structures are built. So
but that's very abstract. You have experimental evidence to support this.
This is evolutionary theory. No, we don't have experiment that is like built in a dish. No. Okay. So we have, so far we are trying to collect data from ecological studies and feed them with our models.
My second question, do you accept that model combine the damage model and the program model? So it seems like they maybe exist at the same time and together to determine the lifespan, right? Both damage and the programming.
I believe no. So I believe in damage, of course, but again, I believe in damage in any other disease or condition. So, and nobody is trying to cure diabetes with damage or entropy in mind. And I think aging in this respect is somehow standing aside from all the other healthcare problems, because there we believe that damage is an issue. I think that just in all the other models, all other diseases, we have good models from the beginning. Damage is there, but we don't care about it. In aging, we don't have a good model. That's why this damage got inflated and took the vacuum that was left by the absent paradigm.
But perhaps here you want to distinguish the average lifespan of the population and the absolute lifespan, right? Maybe the absolute lifespan is programmed, but the average one is more like due to the damage part.
No, this is like a mortality curve question, right? So I think that the program affects both. Of course, there is component of extrinsic mortality that also may play a role. This role can be for or against longevity, but I don't think that having damage in mind, we can understand more. I think that damage is downstream from a program. So it can be that something is directly damaging us, so some inflammatory activation, or it can be that some repair mechanisms are switched off and allow damage to accumulate. But in both cases, we need to inhibit something and not to repair the damage.
Okay, my last question. You say the long lifespan is beneficial, right? Can you elaborate these on both biological side and the social side?
Okay, thank you for this pass. So the biologically, it's very clear. Longer an animal lives, more progeny it gets. So, and it's never too little progeny for evolution. And just from every mathematical model one can build, longer lifespan should be always better than shorter lifespan. However, yeah, you want to?
You finish first.
Yeah,
you finish.
I have finished the biological part. So the socially, of course, socially aging is a huge damage, right? Because we lose experience. We have a lot of people suffering. I read like something the old lady wrote that as you age, you become invisible. So that males are not interested in you, but also other people see you suffer, and they don't have enough empathy to share with you, and so they are looking through you, right? So of course it's a horrible thing, and of course, like economically, people are invalidized, stop working. It's a burden for medical system. And of course, curing aging would be a big deal for our society. I think making it much better than it is.
Okay, I just want a little bit discussion on the biological part. To my observation, See, mice has a very short lifespan. They can have much more progeny than human, which has a longer lifespan. How you explain this?
Well, this is the life history theory, R strategy, K strategy, pace of life stuff. Yeah, of course, the different components of life history are like evolving in concordance with each other. So if mice have high mortality, they need the many niches are freed every month, so they need more progeny to occupy their niches. Or this is a wrong statement. So it's more beneficial to have more progeny because this progeny will have better chances to find a vacant niche. So, this is already a complex question that can have many interactions of how all these things are working together. So the key thing that the only reason for a shorter lifespan that can be found in literature is the pathogen control theory. So all the other models, they favor long lifespan.
Well, aren't there ecological arguments for shorter generation time, because you can basically expose new genetic variation to an environment faster, you have faster generation turnover, so it would be a fitness effect if you basically have this turnover faster, depending on this R, S, and K strategy, which I also have some memories from my ecology classes. And a related question, so I forgot which is which, you know, the one reproduction that have a lot of like mice and human who tend to have a few, right? But if you're basically in the strategy where you have a long developmental cycle, right, bringing up the next generation and have fewer ones, would you always say is an advantage to basically amplify evolutionary advantage? Because I thought this strategy is all about nurturing and making sure this nurtured generation has a fitness effect and can then by itself propagate efficiently. And, you know, that in that setting, actually, maximizing numbers of offspring is actually has its downsides.
Yeah, nobody said that rapid propagation and our strategy many and rapid. So just there is indeed a covariance that animals that have longer lifespan, they typically have fewer kids, and animals who have more children, they are short-lived. However, if you look broadly among animals, so actually the sparrow is a beast. So it has it matures very fast. It has very high fecundity. It's a typical R strategist. So high metabolic rates and high extrinsic mortality. So all these covariances that classical gerontologists bring to sort of rationalize aging are not working with sparrow. This is an animal that if you fly it in any classic models, they collapse. So nobody said that this is a must, that the animal that has a lot of progeny should have a shorter lifespan.
So along the same line, although human being lifespan increased a lot over the last century, but at least females' reproductive span doesn't increase much. So it seems that, I mean, if you say that longer life, more progeny seems not at least apply for human being in terms of reproductive.
Yeah, I got a question. There are two questions. One question is about evolution of human lifespan, another is about menopause.
Right.
So I think last 100 years, the lifespan increased not because of the evolution, but because of the environment. So we just cured infectious diseases. So generally, humans indeed are evolving very rapidly evolve longer lifespans. Since our separation with chimps, we doubled it, being already very long-lived species, being a small population, small population numbers, which is very impressive. We will probably continue do it and probably double it in the next 7 million years if not find a cure. So the second point is that human females have menopause. You know the grandmother hypothesis that like females are becoming less effective at childbirth because of aging, the health decreases, and this female may not be able to deliver a child or produce adequate child care for a long time humans need. And that's why grandmothers are sort of refocused towards taking care of the grandchildren. So, but I don't think this is this question is very relevant to what happened in last 100 years, because it is just very recent. So the evolutionary responses do not work typically with such a speed.
Okay, and also for the social ants, apart from pathogen-induced longevity change, the more natural ones is that if the queen died, then the workers will go to the dew, and then there will be a pseudo-queen rise, and then they immediately increase the lifespan from months to years. But if you reintroduce the queen, they basically return to the workers, and then they will go back to the lifespan a month. There's no pathogen involved. It's a more natural process, or just social pheromones and their insulin signaling. How do you comment on that? Basically, one individual can increase and decrease the expected life expectancy just because of the social status.
Yeah, so I'm aware of this work. I think these pseudo-queens are called hammergates,
right.
And I often cite it as like a paradoxical thing that is not possible to fit into the damage accumulation models. Because where was entropy and damage, where the animal was short-lived, became long-lived, and then became short-lived again.
Right.
Again, so it's obvious that it is something programmed.
But also probably not really support your pathogen control theory.
Yeah.
And also for your pathogen control theory, I think death after infection probably be beneficial. But there has nothing to do with aging. Also like age-dependent immune system failure, whether that's adaptive or not is really a question. Because even if there's kinship protection, the evolution outcome should be like we have a bulletproof immune system, avoiding any kind of infection. But if it doesn't work, it fails, then we just go immediate death. That's probably the best way to protect our kings, right? So I don't get why we want to age a bit more carry the infectious reagents, and then we die. And that's adaptive. I just quite not get the concept.
So probably so immunosenescence is of your care. So why?
Right.
So this is a little bit more difficult model than what I have presented before. So idea is that we have infections that we cannot control with the aging. So like flu or measles or any other COVID, all the diseases that we can recover of. And there are diseases that we can control with aging, chronic sterilizing ones, syphilis, gonorrhea, HIV. So if we look at the epidemiological dynamics of these diseases, so importantly, we cannot go like bacteria, because if we go like bacteria, killing self in response to everything, then we will die immediately of measles or of common cold. And this will be a waste. But immune system smartly can discriminate between common cold type of things and HIV type of things. And one of these criteria is time of first pathogen encounter. Common cold, all these curable diseases, they are typically diseases of childhood. We get sick, we develop lifetime immunity. So, and these chronic diseases, and this comes from their very basic epidemiological properties. I don't have time to go on this. So, but these chronic diseases are typically attacking hosts in later years. And older we are, more chance is it that what we encounter for the first time is not a common cold easy dizzy, but this is a really something chronic bad. And then, so this can be modeled here. So these red ones are chronic attacks, and red ones are acute disease attack and curable, and gray ones are chronic disease attacks. And so older we are, more chances that what attacked us is chronic. And then this phenoptosis, killing self at older age, becomes evolutionary beneficial.
Yeah, I feel that's the consequence of aging instead of a reason for aging. Sorry, that's my perspective. Thank you.
We predict that this is evolutionary beneficial. So this is a fact that should be programmed because it helps our genes to disseminate.
We can soon continue the debate over the networking reception. So any last questions, maybe from the social science perspective? Or?
Thank you. So, we've been reading a lot about these treatments to prolong lifespan by, flushing out all these dead senescent cells. But are we then, are these treatments, what's your opinion on these kinds of treatments? And are we then exposing ourselves to pathogens that might, might not be good for us because these senescent cells are actually benefiting us? So yeah, your opinion on these treatments.
Okay, thank you so much. So there are two questions. One is about do senolytics work? Second, if this will be a problem with the infections. I will start with the second question. I think the benefit of senescent cells is present only within the course of infection. So if the individual is not infected, if we clear senescent cells, it shouldn't be a big issue. So from the infection point of view. So these cells are probably accumulated there and are just damaging. So about the status in the field, I think the senolytics so far are not that shown to be like prolonging life. I think some of them maybe have like minor improvement for health. But these effects are still minor. So should they work or not according to pathogen control model? I'm not sure because it might be that the cellular senescence within the immune system is very important. And maybe if we target these specific cells, then it will have major effect. Otherwise, like targeting senescent cells in other tissues may be of minor benefit.
Hello, thank you for the lecture. So the telomeres, they're getting shorter and shorter, just facilitating the senescence process, right? But in order to stop aging, what is the counter mechanism? Is it just new cells are being new cells reappear, or what is happening according to your theory?
No, well, this is something that we need to develop, but in general, telomerase, I think, is expressed in many adult tissues, right? Ralph will not let me to lie. So like the stem cells and also lymphocytes, they naturally express telomerase enzyme, which makes telomeres longer. Mm-hmm. So in principle, the body can deal with this issue.
Like telomerase?
Telomerase, yeah.
Telomerase ferment. Mm.
That brings us to the end of the session, but not the end of the gathering, because all of you are invited, no matter whether you signed up or not, to join our little networking reception, which is basically snacks that we provide, where we can continue some of the discussion. But before I end, I also would like to thank the knowledge exchange team of the School of Biomedical Sciences, Heidi, Veronica, Erika, who helped to put this all together. Let's give them a hand of applause. And of course, I thank Tarani and the Sau Po Centre for working with us to organize this. And I think it's been fun, and I hope we all enjoy this. And of course, big thanks to our speaker for sharing his thought-provoking theory, I think in a way that also those not directly working on aging or on age-related diseases we could understand. So thank you all for coming, and please join us for the gathering outside. Thank you very much, Peter.
Thank you very much for having me.